All right, one last video for module 2 just to summarize the discussion we have. So far, we have discussed prokaryotes, eukaryotes. Now, we have discussed eukaryotes in terms of how they came to be or how they evolved. Now, let's take a look at the present day eukaryotes. The eukaryotes in our world today are quite diverse. They are the most complex organisms in terms of cell structure, and there are variations in terms of their structure. So, for example, here in our slide, we have a protista, a member of the kingdom Protista, a single-celled vorticella. So the arrow right there is actually the cell itself with a modified flagella, which it now uses to attach itself to the substrate. So it's like a plant, but not really because it's not photosynthetic. They're single celled, and in this case, it's not mobile, so it stays in the same area, but it also consumes other organisms. So the vorticella cell uses its cell the structure, its evolution of new structures to sense the environment, to trap food, expel excess fluid because they live in a moist environment, and they also try to avoid predators using the structures right here. So the eukaryotes basically had evolved to very unique structures. Now, speaking of unique, this is just an overview of the characteristics of plant cells. So as you can see here, the plant cells have the multiple organisms that we have mentioned during our module 1, like the chloroplast, the the peroxisome. But just to highlight again, the unique characteristics of plant cells are the presence of vacuoles. So these are large structures, usually in the middle of the plant cell. In animal cells, the center organelle is the nucleus, but in plant cells, most of the time the vacuoles are even bigger than the nucleus itself, which occupies the middle space of the plant. Now, there are also other unique structures, namely the cell wall, which provides additional structural support for the plant. You have the plasmodesmata or plasmodesma as well, which are like tunnels or holes in the cell wall, which allows the plant cell, which is multicellular, to communicate with the surrounding plant cells. Now, comparing animal cells and plant cells side to side, the bigger among the two would always be plant cells because they can grow up until to 100 micrometers in length. Plant cells are always rectangular or cubic in terms of shape. On the other hand, animal cells are confined to 10 to 30 micrometers in length and are often round or roundish in terms of the irregularity in their shape. So more specific examples of unique characteristics. So again, plant cells have the plasmodesma or the plasmodesmata. They have chloroplasts and the presence of the vacuole. On the other hand, animal cells have the lysosome, cilia, and yeah, pretty much the centriole. Centriole, by the way, is a uh, modified cytoskeleton which the animal cells uses for cell division. Plant cells do not have that, but they still have the cytoskeleton. Okay, so here we go again. Characteristics unique to plant cells. You can read this on your own, but pretty much this was what I have mentioned in the previous part of the video. All right, so now that we have cells that are complex and can communicate with one another, it opens the opportunity or opens the door to the ability of eukaryotes to have multicellular structures or basically multicellularities. So we have in form of one organism can be composed of multiple or hundreds of thousands of cells, and these cells of similar purpose and structure make up the tissues. tissues make up the organs, and the organs make up the organ systems, and so on. In the human body alone, there are 250 types of cells already, and not even all cells have been recognized or even discovered as of yet. But all of these 250 types have unique structures, they have unique characteristics, they have unique roles in the human body. That's how complex eukaryotes became. So let's try to understand, why is it that way? Why is it that animal cells became um, so many different types, like 250 types just in the human body? Well, that's because eukaryotic cells are capable of something called cell differentiation. So meaning the original cell that are from the zygote, or that is from the zygote, remember the merging of the sperm cell and then the egg cell becomes a zygote. Now that zygote can further differentiate. So when we say differentiation, this is the formation of specialized cells. So from the very generic or the multipotent, so I will be discussing that later, the zygote can differentiate into skin cells, bone cells, or also known as osteocytes, red blood cells, muscle cells, and fibroblasts, which is a part of the um, connective tissues, and also um, nerve cells. So there are so many cells in the human body, but again, they came from one single cell, the zygote, and then they eventually just differentiated into these specialized functions. Now, always the numbers and arrangement of the organelles relate to the function and activity of the cell. So the structure of the cells themselves would give us clue on what they do for the human body. So for example, adipocytes or the fat cells are mostly empty because they simply contain the fat or the lipids in the human body. Bone tissues made up of osteocytes are usually rich in calcium and are closely packed together, which enables the bones to be more rigid and provide a structural support. So all of these cells have their own characteristics and their own functions. Now, what I was referring to earlier when we say multipotent or pluripotent, these are embryonic stem cells. So you might have heard of the term stem cells before. When we talk about stem cells, these are special structures or special type of cells wherein they are able to differentiate into multiple types of cells. So let's take a look, for example, of the embryonic stem cells, the original cells from the zygote that you were in the beginning of your conception. So these stem cells, these embryonic stem cells, are capable of becoming any human cell. It can be your skin cell, it can be your fat cells, muscle cells, and so on, because they contain the information and the ability to differentiate into specialized cells. That's what we call as pluripotent. Now, usually, as the term suggests, embryonic, is only found in embryos. So it is an amazing cell, but it is confined in the very early stages of our human development. Now, in our human body, in the adult human body, there are still cells that are multipotent, meaning they can still differentiate into different types of cells, but are quite limited. It's not like embryonic stem cells, which have unlimited potential. Multipotent cells can only differentiate into specific types. So, for example, adult blood stem cells are found in the bone marrow, and they can differentiate or divide into becoming red blood cells and also white blood cells and lymphocytes and so on. So, limited but still amazing when it comes to the multipotent cells. So these are just examples of the different animal tissues. So for example, slide A, I hope you recognize this. This is a red blood cell with some white blood cells here in color violet. And then we have the muscle cells here, which are striated, meaning they have this parallel lines because the muscle cells can contract and extend. Next, you have here letter C. These are skin cells. So notice how compact and flat they are, because again, their purpose is to provide an outer layer and protection. And then lastly, at letter D, we have the nerve cells. So you can see the central cell with multiple extensions to pass down or to pass the signals from one cell to another. So as you can see, they're all cells. They have different organelles, but in terms of structure, they have differentiated based on their function. Okay, so these are just some more details of the other types of cells that can be found in animals. All right. So knowing all this, where does the science take us? So what is the current trend? So we have, humanity, been exploring the concept of tissue engineering. So basically, we do not harvest cells anymore from other organisms. For example, um, if we are looking at organs. Organs are very limited in terms of number, and sometimes they would require um, donors. For example, a heart donor or a bone marrow donor or a blood donor. So sometimes these are quite difficult to understandably to secure, because that would require some sacrifice or end of another human's life. So to answer that predicament, what if we could just engineer our own? What if we could just create new blood cells from living cells? What if we can just create our own blood cells? What if we could just make our own heart and liver and so on? So that is what tissue engineering is. We're trying to create new, like, human parts, because to answer the need for them in the medical field. But biocompatibility is the major challenge in artificial organ development. So that means your body should not reject these engineered tissues, because it has to be compatible. And that's like one of the major things that we have to check whenever we have transplants. So, for example, your blood type has to match, and your DNA must be quite similar, so that your body will not detect the donated organ as a as a foreign entity and start attacking it. So for example, you are blood type O, and for some also scratch that. For example, you are blood type A and you were transferred blood type B. Your own immune system will start attacking the transferred blood, which puts your life at risk, basically, and then eventually leads to death. That's what we are trying to avoid when we are developing our own artificial tissues and cells. Biocompatibility. So the strategies to overcome this challenge is for tissue engineering to improve our own cell culture. So for example, we could, yeah, to scaffold, like we have a 3D pattern already, and then we'll just scaffold these cells there. So for example, we have a scaffold for the human heart, so that we could create the heart, basically let the cell divide and shape into a heart. And that's what we use in making replacement organs, so something called as organoids. So in this case, the concept is to allow the cell, or allow the organ to self-assemble by making the parts, if you can imagine that. But is this still possible as of now? Not really. It's very expensive. What we have found so far is cultivating our own organs in the laboratory is way too expensive than just looking for a donor. That's why it's still quite limited, and we have not perfected it. It's still very difficult to create new cells. To create new cells and new organs, it's more difficult than we have anticipated. That's why it's not yet possible as of now, but let's see if we will see, or we will be encountering more breakthroughs in the medical or scientific community. Okay, so that's that. That's the end of module 2. I hope you learned a lot from our discussion. And again, if you have any questions, feel free to message me in MS Teams or talk to me during our class session. Bye, everyone. Take care.